So, hi, good morning, everyone. Am I audible? So, all is well. So, as we discussed in this class, I am going to discuss about some of the very important but very required basic terminology where students usually struggle, they don't understand in a very clear manner, right? So, and I am starting the entire things with a small question. Just try to answer it. If I ask a question, duplication of DNA occur in which phase of cell cycle? I'm giving you option also to make your life easy. First option is G2 phase. Second option is S phase. Now it's all right? All right? Now it is audible? Yeah. Look. So very good morning to everyone. So as I committed, in this class we discuss about some of the very important and basic terminology where students struggle usually. And definitely trust me, after this class you will not struggle. So listen it very carefully. Ask your question where you are finding you have certain difficulty. So I am starting the story with a question. And the very first question I wrote here on board is, duplication of DNA occur in which phase of cell cycle? This is the very first question I have written. I have given the option G2 phase, second option S phase, third option NA phase, fourth option NA phase 1. You are voting for which answer? Many people wrote, which one? Once again, second answer. And answer is second, S phase. And another question I asked here, and this is the 2023, this year NEET question. If you recall, you will find. So that the question is, replication of DNA occur in which phase? And same option I have given. What the answer will go for it? So here answer is differing from student to student. Some of the student is saying correct answer is S phase. Someone writing 3. In both the question, answer is same, S phase. The meaning of replication of DNA or the meaning of duplication of DNA are same. Many times students confuse what? Duplication of DNA, chromosome number double, and that occur in anaphase. So usually many students make mistake in the first question. Okay, they just misinterpret duplication of DNA as doubling of chromosome, and they just confuse and they just write the answer as anaphase. But both the question have the same answer, that is which page? S page. Got the idea? So this is the answer of the question. Now I am going to tell you the concept behind it, terminology behind it. I hope many of you are struggling with some of the terminology. One word I am writing gene. Another word I am writing a DNA. Another word I am writing chromatin. And in chromatin I am writing 2 different chromatin. euchromatin. Another I am writing heterochromatin. Then I am writing chromatid. Then I am writing chromosome. Then I am writing centromere. Later on I am writing kinetochore. And further I am writing about which one? Telomere. These are the terminology required for understanding of cytology also, cell biology also. You can say about cell structure, cell cycle, and genetics. These all terminology is going to use in these 3 different chapters, and it is very required you have a very clear basic understanding about it. You must have a very clear basic understanding about it. So do you find you require some support here for basic understanding, and all of you are finding no, I am pro here, sir. say something else. So I will not waste your time. I will ask one or two questions and then we will move ahead. So what you feel? You require here the explanations, or you find like, no sir, I am very comfortable. Someone writing, sir, yes, need understanding. Okay, explanation, right. Okay, ji. Have a look. Now give your attention. So before moving on, to connect you people better with the concept, I am asking some small question. Got the idea? I am asking some small question. So the very first question I am asking is, how many genes present in a DNA? So who voted for no, especially I am requesting to you, please try to give these answers. So I also get help if from your side it is all well. Let me write all the question first and then we will see toward the vote. So definitely I can find how many of you are answering very correctly. So first question, how many genes are present in a DNA? How many DNA present in a chromatine? Next question, how many chromatin is present in human diploid cell? Next question, you note this question, it will definitely going to help you a lot. Please, I am requesting all of you, note this question in your notebook. How many DNA present in a chromosome? How many DNA present in a chromosome? Getting the things or not? Next question, what is the difference between chromatid and chromosome? Chromatid and chromosome. I am asking one or two more questions, and all these questions going to solve easily after discussing about these things. Another question I am writing, how many centromere present in a chromosome? Next question, how many kinetochore present in a chromosome? So these are the some of the questions I have written on board, and because we are not looking on each other, so it may be a time lag came. So I am asking question by question and answer the question for what I am asking only. So let me come on the very first question. So I will manage to know, are you giving the answer of each particular question? So you just think like, I'm just talking about, I'm giving this numbering of the question as first question. So dear children, you tell me what the answer you will opt for the first question. Now I am looking toward the screen. Question is, how many genes are present in a DNA? Please give your answer. Someone writing answer as 1. Some student wrote answer as 30,000. Some had wrote 20 to 30,000. Someone have written 20,000. Someone have written 2. So these are the different variety of answer I am getting. And one answer came, sir, it varies, sir. Varies means different from DNA to DNA, you want to say. If I am correct, I am interpreting you correctly. It varies, sir. One answer came, it varies, sir. Right. Okay. So, except one answer, every answer is wrong. Except one answer, every answer is wrong. And what is the correct answer of the question? I will explain it also, don't worry. Question is how many genes are present in a DNA? So answer of this question is, it is variable. Depend upon chromosome to chromosome. It is variable. Depend on chromosome number as well as species. It is the first chromosome or second chromosome of which species. So one student told differ in organism to organism. It is also up to certain level, it is correct answer. I will decode it in much detail. Don't worry about it. This is the agenda of the class, because when I teach in the class, usually I find students struggle in this particular area. Understanding the idea or not? So definitely today I am going to decode all these answers, and definitely you will not got any problem after this class. Right? So have a look. So answer first I have given to you. Now first I want to know about your input. So it will help me to think about how much, up to which degree I have to discuss in this question. Next question I asked, how many DNA present in a chromatin? Second question you just gauge it. Suppose that it is question number 2. Now start voting for question number 2. How many DNA present in a chromatin? So most of the people are voting for 1. One student wrote 146 base pair. Right? Someone have wrote 200 base pair. Right. Someone have wrote, sir, 46 DNA molecule. But majority of votes are in the favor of 1. And no doubt, most of the students are correct here. What is the correct answer? 1. I will also explain it how 1. Got the idea? I will give the entire explanation for every answer. First, I just want to know your input. And of course, after that, I will explain it. Then my turn will come. Okay? It's all right? Next, if we talk, how many chromatin is present in human diploid cell? Then what the answer you will give? You think like it is a third question. How many chromatin is present in human diploid cell? So many answer coming in the favor of 2. Many answer coming, I think mostly people are writing 2. Some student wrote 1. Some student wrote 4. Some student wrote 146 base pair, right? So these are the different answer is coming. I am sorry to say here all of you are wrong. Got the idea? The question here is how many chromatin is present in human diploid cell? The correct answer of this question is how much? 46. I will explain it also. Right? The correct answer of this question is 46. I will give you the explanation of this question also. Right? Next question what I ask. How many DNA present in a chromosome? How many DNA present in a chromosome? What the answer you will give for this question? First, your input is important, then I will give my input. How many DNA present in a chromosome? Assume it is question number 4. What answer you want to give? One student writing 1, another writing infinite, someone writing 2, someone writing 23. So these are the variations of the answer coming from your side. Is it all right? So I am giving the correct answer for this question. How many DNA present in a chromosome? It is either 1 or 2. Depend on stages of cell cycle. Stages of cell cycle. If it is coming like option is there 1, you can choose 1. In some stage of the cell cycle 1 is there, in some stage of the cell cycle 2 is there. We will also talk about it. Got the idea? Now we will talk during teaching what is the difference between chromosome and chromatid, because it is a subjective question. So I am not expecting from you, you write the answer of the question. Definitely I will explain it. So we are not going to asking about this question right now from you. Now another question is there. You can say in the one word, how many centromere present in a chromosome? How many centromere present in a chromosome? Here you are definitely giving a very impressive answer. Very good, well done. Most of the students have given the correct answer, and the correct answer of this question is what? Basically 1. Well done, very good. So how many centromeres are there? One chromosome have 1 centromere. Got the idea? Now another question. How many kinetochore present in a chromosome? Here also I am getting an impressive answer by many of the students. Well done, very good, well done, well done, well done. Most of you except one student have given the correct answer, and that is what? Basically 2. Got the idea? Now we are going to deal it about in much detail. Have a look here. Give your attention. So here you better know about DNA. So have a look here. This is a DNA. Double helically coiled structure. This structure is a DNA. DNA have 2 strands, and it is a which one? Double helically coiled structure given by whom? Watson and Crick. Okay, ji. So this one is a double helically coiled structure. I'm not decoding the DNA right now. So how many DNA is this? You will say for this one, how many DNA? 1 or 2 DNA? Say fast. It is 1 DNA or 2 DNA? It is 1 DNA. Here it is 1 DNA. Very good. This is 1 DNA. Done? Clear in the mind? Now I am asking how many genes, what is genes basically? Gene. Gene is functional, I am writing a single liner definition right now. Functional fragment of DNA. Gene is what basically? Functional fragment of DNA. Right? Clear? Functional fragment of DNA means that much bigger fragment who can control a function, who is responsible for a function, who is responsible for a character is called what basically? Gene. Now, if I ask a question, suppose that this is a DNA, this is a first DNA of a human. I'll not talk about the species, otherwise you will unnecessarily confuse. Suppose that this is a DNA. So how many genes are present on DNA? On this DNA. So it's depend on a species or it's depend upon chromosome. It's depend upon the DNA. Suppose that this is one DNA and this is another DNA. This is another DNA. So it may possible that length of 2 different DNA present in a species is variable. It may possible the length of 2 DNA present in a species is variable. I am assuming as a human being. If I am asking a question, use your brain and answer it. In your body, which chromosome have the longest DNA? In human body, which chromosome have longest DNA? Chromosome number 1. Very good. You people are really doing great. Impressive answer is coming. And second longest DNA? Second longest DNA present in which chromosome? In human being. Second longest DNA present in which one? So second longest is present in which one? Second chromosome. Usually, if you not talk about few exceptions, we give the chromosome numbering based on the size. Longest first, second longest second, third longest third, fourth longest fourth. But I use a word if we not talk about few exceptions in human karyotyping. So this is one DNA, this is another DNA. So in this 2 DNA, number of gene may be variable. Suppose that this segment is a gene. This is another segment, it is another gene. This is another segment, this is another gene. This is another segment, this is another gene. And here suppose that, so how many genes we are finding here? Assume it is 1 gene, 2 gene, 3 gene, 4 gene. Getting the idea or not? So here suppose that I am just telling you, I am just trying to make you understand as a layman. Suppose that here to here 1 gene, here to here 2 gene, and here to here 3 gene. 1, 2, 3. Here it is which one? 1, 2, 3, 4, 5. So what I want to convey you, number of gene on a DNA is what basically? Variable. In this chromosome, different number of gene is there. in this chromosome, different number of gene is there. And if it is a first chromosome of X species, and if it is a first chromosome of Y species, in that case also number of gene will vary. In same first chromosome, in X chromosome, in this first chromosome have different number of gene, and in this Y species, in this first chromosome, different number of gene. And that is the reason I told you one thing. What? The number of gene on a DNA is variable. It varies from DNA to DNA, and it is also decided by it is the DNA of which species. See, some genes are longer, some genes are shorter. Got the idea? So number of gene length also going to vary. Able to understand? So length of gene will decide, length of DNA will decide. So what the answer is coming, what the best answer of the first question is coming. I asked here the very first question. What the question I asked here? How many genes are present in a DNA? And here I have given a answer. It is variable and depend on chromosome number or DNA number. Chromosome number means DNA number, usually we not told the word, as well as which one species. Picture came in the mind or not? Picture came in the mind or not? Are you able to decode the answer of the first question? How many genes are present on a DNA or chromosome? It is variable. It depends upon which DNA and which chromosome is there, or it depends upon which species is there. Understanding the idea? Gene is nothing. Gene is simply a fragment of DNA. Gene is simply a fragment of DNA. Now I am coming on the next thing. Have a look here. Give your attention. Now this is a DNA. DNA. Now I am asking a question. What is the difference between DNA and a chromatin? One student asking, sir, any weekend exam? No, not at all. Because these are introductory things we are dealing with. Tracks it will start from the Monday. So just simply in these class, we are just trying to understanding each other. So what benefit will occur? Somehow I will understand because you are not present physically. So you will understand my style. I will understand somehow you. So you just think like these are the some of the basic understanding we are developing between each other. Is it clear in the mind? Right? One student asking, is gene participate in translation? Of course it participate, and I will teach you in coming day in much detail how gene participate in translation. Getting the things, have a look. So it is a DNA. Just I am asking a question, what is the difference between a gene and a chromatin? Any picture you want to give me? One student asking, till when YouTube classes going on? See, it is initially it is going on because admissions is in process. As the student get settled, definitely we will shift on the app, because right now I can't give the someday some X is coming, some Y is coming, someday some X is coming, like these things. Flexibility is there. So after the things become stable, definitely we will provide you the app ID, and we will add you people in the apps of the Sri Gosalite. Okay, right. One student writing, DNA condensed to form chromatin. DNA condensed to form chromatin. How many of you are agree with it? And now question arises, no weekend this week. See, from Monday onward our track sheet will start. So when Monday onward till Saturday, It is a introductory class. We are understanding each other simply. We are just talking, gossiping, chit-chatting, right? And the real track sheet will start with which one? Monday. Because in your physics chemistry, some basic physics is there, some basic calculation are there. So they are taking about these basics area, which will help you in the which one? The part of mathematics like calculus and all, it will help you in which one? To understand the which one? Physics better. So likewise here, what in biology? So we are just telling you about some basic terminology where you are just going to struggle. Got the idea? Picture came in the mind? Right? It's all right. So when from Monday tracks it will start, Monday to Saturday we will teach you, and Sunday we are going to conduct the exam. So the examination going to start as your main tracks it will start, and that going to start from which one? Monday. Is it very clear in the mind? It's all right. Can we come on the topic? Yes, sir. Please tell about the new NCERT edition. See, something you trust on us and leave on us. And I think it is a better idea. You trust on us and you leave on us about the new NCERT. Look here, what is the problem? I am telling you. Many students are asking question. What to do, sir? Read old NCERT, read new NCERT. Don't be panic with these small thing. Don't be panic with these small thing. Do you know, I am telling you a very honest thing. I get panic in last class. Do you know the reason behind it? Anybody can tell me what was the reason behind I get panic in the last class? If you just find like I was panic in the first class. Do you know what is the reason? Do you know the reason? If you attended the first class, I just want to check how many of you understand the reason of my panicking in last class. Right? I am telling you what was the reason of panicking. I started discussing the question paper. The first question I opened, due to typing error, no answer was coming. Getting the things, due to typing error, you just find like, na? Then I told in the last, it is a typological error. In place of snRNA in third option, they have wrote 5.8 sRNA. I am solving the question, you are solving, not answer. Every answer is looking incorrect. Now open the NCERT, either I am wrong. So check, no, I am not wrong. So then nobody have the all option in the mind. Na? Then I also told you, check your paper, some missing is there, something wrong is there. Then I went, see the question paper, I find it is not 5.8 sRNA in one question, it is which one? snRNA. Okay, it happened. I find like just few days before exam occur, we have given the answer key. Now I am solving the paper, no answer is coming. Getting the idea or not? So what I want to convey you, just try to understand. About new NCERT, about old NCERT, you please leave it on us. Why I am telling you don't be a part of it? Till now, no one have any idea. Till now, no one have any idea. If you ask anyone, sir, what exactly going to ask in the NEET exam, how anybody can say? Until and unless NTA, NMC not give any guidelines about the syllabus, how anybody can say? We know what chapter is added, we know what chapter is deleted. So what we have planned already, कि first we teach you that topic which is also in the new NCERT as well as in old NCERT. Later on in the last, we will kept that chapter who are not in new NCERT. An example in plant physiology, transport of plant and mineral nutrition. These two chapters not in which one? New NCERT. So we placed it in the last in the track sheet. Keep up to the last, any time in between, NTA will give the notification. Na? These are the syllabus we are going to ask. So if they will keeping this topic, we will teach it. If they are not keeping it, we will not teach it. So these things we will take care. Got the idea? Okay, so you don't be panic. You just simply focus on your work. And what is your work? I am telling you, what is your work? See, you are just taking one more attempt in the life. Got the idea? So that time, this time, two things we are expecting from you people. One, don't do procrastination. Second thing, I'm just telling, do you know what is the reason behind not getting a success? One student asking, sir, Monday, which chapter you are teaching? Of course, we will start with very factual chapter. And that is what? Morphology. That is what? Morphology. And there is also a reason behind it. Why? I am telling, suppose that some student coming 2 days late. So morphology they can manage because mugging up is more. If we start with a big conceptual chapter, what's happened? The student will suffer, they will struggle. Getting the idea or not? So that is the reason in the beginning, at the time of the stabilization of the batches, we are starting with a such kind of chapter. If happen, some student are finding, I will just go to campus 1 day late. Even in online, okay, I'm thinking what to do. I will go for the procedure of the admission 1 day late, 2 day late. So that scope they remain, they have ki if it is a factual area, okay sir, route what is big, what sir will do in the route, I will take care of it. So that is the reason we are starting with some easy nature of chapter. Getting the idea? Timing is same. What timing going right now, same timing will be there from the Monday onward also. There is nothing a change. Okay. After morphology, anatomy. Of course, that sequence will move. Okay. So have a look now. Just give your attention. Please come on the agenda. So what I am telling, I am expecting this year two things from you people. If you give two commitment, we can commit your result. Two commitments simply we want. One commitment is don't procrastinate. Procrastination, you know, is a beautiful word of the English and it is very well applicable in the student's life. Do you know the procrastination? Meaning of procrastination? Shifting the work from today to tomorrow, postponing the work. Postponing the work. Today, okay, today I am busy in this, tomorrow I will do. Tomorrow also some work came, day after tomorrow I will do. Got the idea? That is procrastination. Fix the work. Second thing, are you aware about Parkinson's law? Are you aware about Parkinson's law? After the class, you search on Google, what is the meaning of Parkinson's law? Got the idea? I am not asking Parkinson's disease. I am asking Parkinson's law. Parkinson's law say that the work spread in the time allotted to do that work. Should I explain it? It is a very important thing which will apply in your academics or study. If you handle these two things, no one stop your success. I am telling you. I am just giving you a small example. Try to understand. One is a housewife. Another is a working lady. Amount of work in both the house are same or different? Almost, do not gauge in exact way. Amount of work, nature of work in the house of both working lady and housewife is same or different? Amount of work. It is almost same. Why? In every house, cleaning require, making breakfast, lunch, dinner require. So nature of work and amount of work is almost, you are eating, you are cleaning your house, cleaning your clothes, getting the thing. So nature of work and amount of work are same. Now you see a working lady and also see a housewife. Working lady also doing all the job, but she have a target. I have to finish this work in that much time only. Many people not afford the servants. Their salary is not that much. Getting the things or not? So they are just doing same work in the allotted frame of time. I have to start for my office from 9 o'clock. So before 9 o'clock, they have a target. I have to finish my job. A housewife. She is not going out. She is housemaker. She is also not sitting. You will not find your mom if she is housewife, she is sitting in the home. She is also working whole day. Means what happening? She have somehow bigger lap of time. Work spread in that particular time because she is mentally what? Okay, this is the time now. So what happened? The speed of work will slow, and that is called Parkinson's law. What the time is available for you for any work, your work will take all the time. Means what I want to say, how it will apply in your life. If you want to finish an example, if you are revising, you are giving a target. I will revise morphology. An example I am telling morphology because very first we are going to deal this factual area. I will revise this morphology in one day. It will take your entire day. If you given a target, I will revise the morphology in 2 days. It will take your 2 days. All 2 days will consume in morphology. You are not finding my time is going. If you want to revise morphology in 3 days, it will take your entire 3 days. And if you have given a target, I will revise the morphology in half hour, you will also do it in half hour. So amount of work going to occur is same in half hour also and 3 days also. Means always give yourself a tough target, impossible target every day. Your brain will make it possible. Your brain will make it possible. Getting the things or not? Right? Clear in the mind? Is it all right? Understand the Parkinson's law? So always when you are studying, give yourself an impossible target. Today I will finish this thing. Right? One student asking, sir, for girls also, is morphology start in the first week? Yes, in both boys campus and girls campus, same tracks it will run. Got the idea? Picture came. Now we are coming on our topic. Have a look here. What we started. So now look. Now look. So don't procrastinate. Focus on the class. Be disciplined. Self-discipline. And of course, for online batches, self-discipline is very compulsory. Now look here. Now how chromatin is different from the DNA, I am telling you. This DNA is a long, and this DNA is going for packaging. And during packaging, what's happened? This DNA wrapped around a octamer of protein, histone protein. Who is wrapping behind? Around? DNA is wrapping around the protein octamers. Got the idea? Let me tell you. Suppose that this is DNA and this is histone protein octamer. DNA is wrapping like this. DNA is wrapping like this. This DNA is a long DNA. Otherwise, you will feel like, sir, you make the chromatin bigger than DNA. Suppose that this is a DNA. Getting the idea or not? Longer DNA. These are longer DNA. and these DNA wrapped around, it underwent in condensation, it wrapped around a histone protein octamer. This structure, yellow color, is which one? Histone protein octamer. This is histone protein octamer. Is it clear in the mind or not? The structure who formed here is called chromatin. So you got how chromatin and DNA differ from each other. And if I ask a question from you, tell me, dear students, in one chromatin, how many DNA is there? In one chromatin, how many DNA is there? What the answer you will give? In one chromatin, how many DNA is there? This is the DNA, same DNA. You don't think like you make one strand here. Otherwise, many people also confuse, sir, you are making only one strand. I am making two strands. Don't worry. It is both strand is there. Okay, it is not single strand. I just, okay, getting the idea. So, how many DNA is there? How many DNA is there? One DNA is there. Picture came in the mind? Understanding came, clear or not? So this structure is called chromatin. And here so many beaded structure is there, na? It is like bead like a structure, are you finding or not? Tell me. These bead like a structure, yellow color wrapped with the white color, are you finding in the diagram or not? So each bead like a structure is called nucleosome. Each bead like a structure is called nucleosome. So can I say things कि many nucleosome together forming a chromatin? Can I say chromatin is made up of a repeating unit of nucleosome? Yes, na? Many nucleosomes are there. Repeating unit of nucleosome is going to form which one? Chromatin. Picture came? Understanding came? Is it all right? Can I move ahead? Yes. So DNA, chromatin, how it is differing from each other, and here it is histone protein octamer. One bead structure is called nucleosome. I am not going to talk right now here in one nucleosome, 200 base pair of DNA is wrapped. I am just focusing on what I want to convey you today. Now have a look. Now this chromatin is undergoing in further condensation. Here also condensation occur. And here it is also going for further condensation. And when it is going for further condensation, who is involving? In this condensation, which protein is involving? Histone protein. And here who is involving? Non-histone protein. And in this further condensation, what's happened? You have a look. It becoming like this. So this longer one condense and become like this. After condensation, it becomes shorter and thicker. It becomes shorter and thicker. What this shorter and thicker structure is called? What this shorter and thicker structure is called? This shorter and thicker structure is called chromatid. Now I am asking same question. Or it is also called chromosome. It is also called chromosome. Getting the idea? Now, I am asking, in this chromatid or chromosome, how many DNA is there? One DNA is there. DNA was this one only, na? This is the only DNA becoming after folding like this. Same DNA. This is the DNA. Get folded and become like this. So in this chromatid and chromosome, how many DNA is there? One DNA is there. Clear? Picture came in the mind or not? Now, what is this? Here a constriction you are finding, na? Thickening is uniform or here less thickening is there? Less thickening is there, na? Are you finding in the center? I am finding what? It is less thickening is there. Clear or not? Is it all right or not? So this less thickened area is called primary constriction. It is called which constriction? Primary constriction. And another name of this primary constriction is called centromere. Centromere. How many centromere is there? How many centromere is there? One centromere is there. So for your better understanding, I am putting a dot by which you will understand the centromere. So how many centromere is there? How many centromere is there? One centromere is there. Clear in the mind? Is it all right? One centromere is there. This side and this side, both sides of the centromere, it is arms of the chromosome. It is arm of the chromosome. Are you getting the idea? This is a arm of the chromosome or chromatid. This is a arm, and at the tip, here it is called which one? Telomere. Here it is called what? Telomere. Picture came in the mind. Now you will say, what is the kinetochore, sir? I am drawing the diagram also. Here you will find there is a disc-like structure in the both side of the centromere. Here. This is called what basically? Kinetochore. Picture came in the mind or not? Is it all right? So clear in the mind? Now you will ask a question to me. Usually a student asks, sir, in my knowledge, what I have studied, according to this, in my mind, picture of chromosome is like this. Since childhood, I am studying the chromosome like this only. I think it is your imaginary chromosome, na? Since childhood, probably people are studying which chromosome? This is the chromosome. Getting the things or not? And you are telling me this is the chromosome. I am saying this is a chromosome. And your mind have an imagination, this is a chromosome. Then how this is differing from each other? What are the difference between this? So if it is a one chromosome, is it a two chromosome? If it is a one chromosome, is it a two chromosome? How many chromosome is this? I am writing A and I am writing here B diagram. In A diagram, how many chromosome is there? In A diagram, how many chromosome is there? One is there. And in B diagram, how many chromosome is there? In B diagram also one is there. Mostly people writing B also one chromosome. Some student writing no, in B two chromosome is there. So in both A and B diagram, number of chromosome is 1. Here, number of chromosome equal to 1. Here also number of chromosome equal to 1. Here number of chromatid, help me. Here number of chromatid equal to 1. Here number of chromatid equal to, help me, 1 and 1, 2. Here number of DNA equal to 1. Here number of DNA equal to 2. Here, if amount of DNA is C, here it become 2C. Got the idea? This condition is called monad. This condition is called monad, and this condition is called dyad. You make a thumb rule in your mind. Here I am writing number of centromere is 1. Here it is also number of centromere is 1. You make a thumb rule in your mind. And what is the thumb rule? You write as a note that the number of chromosome is always equal to the number of centromere. Number of centromere. Getting the idea or not? So have a look here. In this diagram, how many centromere is there? And in this diagram, how many centromere is there? In both diagram, 1 centromere is there, na? So number of chromosome is equal to number of centromere. Make a thumb rule, you will not confuse. If I am asking a question, look here. Wait a minute. How many chromosome is there? 1. Because how many centromere I made? All chromatid join with common centromere. So if centromere is 1, how many chromatid is there is doesn't matter. Number of chromosome will be how many only? 1 only. Picture came in the mind or not? So here it is a chromosome with single chromatid. And during S phase of cell cycle, this structure of chromosome converted into this type. During S phase, monad converted into which one? Dyad. So when a chromosome have single chromatid, that condition is called monad. When a chromosome have 2 chromatid, that condition is called dyad. So B1 is a dyad, A1 is a monad. In A and B, I have written the differences. Are you able to understand or not? Let me just going to minimize it. Then question as well as everything will come. Are you agree with me, na? I have written number of chromosome is 1. Number of chromosome is 1. Why it is 1? Because this one also have 1 centromere. This one also have 1 centromere. So number of chromosome is 1. It is justified or not? It is justified. Now see, I wrote number of chromatid is 1 here. This is the chromatid. Here 2 chromatid is there. One chromatid is this. Another chromatid is this. How many chromatid came? 2 chromatid came. Understanding the idea? 2 chromatid came. Here number of DNA 1. This is what? 1 DNA. Here how many? In S phase DNA replication occur. In S phase what happened? Suppose that this is a DNA. In this DNA under S phase making another copy of DNA. So it is called what? DNA replication occur. Getting the things or not? And when this coiled with which one? Histone protein, it is becoming like this. This coiled with histone protein, this is becoming like this. But what happening? They are not separate. They are joined with which one? They are joined with the common centromere. So in a space, these are the story which is going to occur. They are joined, both the chromatid going to join with the common centromere. So number of chromatid become 2, but number of chromosome remains same because it is the centromere. Centromere is the kitchen of the family. I always say, 2 brothers are there, residing in the same home. When you say brother are in a joint family and brother in a nuclear family, they are separate or they are in a combined family. When you are saying brother are residing in the same house, two brother family, you, your mom, your dad, your brother, sister, your uncle, his wife, their children residing in the same home. When you are saying we are in a joint family and when you are saying we are in a nuclear family, means uncle and father is separated, and when you are saying uncle and father is together, answer the question. Someone saying wrote something I have not seen. Reply, sir, for what you are asking for reply, I have not seen your messages. I was busy that side. Sir, are these classes free for entire year? No, dear children, these classes are not free for entire year. These are some basic initial classes who are free, and after that you can access these classes whenever you can join it, right? Okay, ji. So I ask a question here. When you are saying your father is together and father and uncle are together and your father and uncle are separated, tell me the answer of this question. It is an answer of question of social science. It is a question of social science. When together, when separated? When two family are together and when two family are separated? Tell me, hurry up, fast. Answer of this question is not coming. Really surprised. You have not seen a joint family and nuclear family. You have not seen such kind of things. Together, what is the proof they are together? And what are the proof they are separated? If you are residing in uncle, mummy, auntie residing in the same house, when you are saying families together and family are separated. Yeah, Nabe, I have given a very good answer. If cooking in the same kitchen, cooking together, it is called joint family. Means who is connecting the family together? Kitchen. Getting the things or not? Sometimes what's happened? One student residing in the same house. Sometimes not happen for every people. Okay, we get separated, we are going in different home, you are going in different home. Because purchasing a home require a lot of money. So when two families segregated, what separated? Kitchen separated. So kitchen separation is showing that they are separate now. Your auntie cooking different food for their children, your mummy cooking different food for you. They are not cooking together. Beside of that, they are residing in the same home. It is called what? Now family is separated. So kitchen is joining the two family. Likewise, here who is joining the two chromosome? Who is joining the two chromosome? If it is a story like this, I am telling you, wait a minute. I am asking a question. How many chromosomes is this? How many chromosomes is there in this diagram? 2 chromosomes. How many centromere in this diagram? 2 centromere. 1 centromere this, 1 centromere this. You have studied about it in metaphase stage. Got the idea? Understanding came or not came? Now, I am making another diagram. This is anaphase. How many centromere is there? How many chromosome is there? 4. Got the idea? So whenever two sister chromatid join with the help of which one? Centromere, common centromere, they are called one chromosome. Look here, na? Here one centromere, upper and lower chromatid, beside of 2 chromatid is there. How many chromosome is there? 1 chromosome. As they divide, they move up, they move down. Centromere also divide. This have a separate centromere, this have a separate centromere. They are considered as 2 chromosome. They are considered as 2 chromosome. Total how many chromosome? 4 chromosome. So who is working like a kitchen in chromosome? Centromere. Understanding the idea or not? And that is the reason I have given you a thumb rule. And what is the thumb rule? Number of chromosome is always equal to number of centromere. Clarity came in the mind? And in one chromatid, number of DNA is always 1. And in a chromosome, number of DNA either 1 or either 2. Picture came or not? So I am telling, is it a chromosome or not? Is it a chromosome? How many DNA is there? How many DNA is there? 1. Is it a chromosome? Is it a chromosome? Yes. How many DNA is there? How many DNA is there? Answer it. 1 DNA, 1 DNA. Total how many DNA? 2 DNA. So what is the number of chromosome in a, number of DNA in a chromosome? Either 1 or 2. It depends on stages of the cell cycle. When chromosome in monad stage, it is called monad stage. It is how many? 1 DNA. When chromosome in dyad stage, which stage? Dyad stage. How many DNA? How many DNA? 2 DNA. But a chromatid always have how many DNA? 1 DNA. Now I asked a question, in human being, how many chromatin is there? Look here. This is a human cell. And this is the human nucleus. I ask, in human diploid cell, how many chromatin is there? I am asking a question, in a human diploid cell, how many chromosome is there? What the answer is there? In human diploid cell, how many chromosome is there? What the answer you will give? If I remove a cell from here, how many chromosome will come? 46 will come, na? So this is a chromatin. This is a chromosome. 1 chromatin converted into 1 chromosome. Understanding the idea or not? 1 chromatin converted into 1 chromosome after packaging. Understanding the picture? Now what happened? In your cell, in your nucleus, this is chromatin 1, this is 2, this is 3, this is 4, this is 5, this is 6, this is 7, 8, 9, 10, 11, and likewise it is how many? 46 chromatin is there. How many chromatin are there? 46. And when they condense, it make how many chromosome? 46 chromosome. Understanding the idea? Now I am asking a question. How many network of chromatin is there? This structure is called chromatin network. So network is one, na? Network is one. But in network, how many chromatin is involved? 46. Because later on it convert into how many chromosome? 46. Picture came or not came? Understanding came or not came? Now, I also use a term euchromatin and heterochromatin. I am just giving you a basic idea. Look here. So this less coiled part of the chromatin is called euchromatin. And this densely packed, tightly coiled region is called which one? Heterochromatin. I just want to give a picture in your mind. Later on when we will teach, we will discuss so many differences between euchromatin and heterochromatin. Understanding the idea or not? So this is the loosely arranged chromatin is called euchromatin. Densely arranged one is called heterochromatin. So I hope what the agenda we started in the today class is successful. Now tell me anywhere you are missing, definitely I will help you out. So we started with some of the terminology. I hope I am just going to clarify. Many of you know from earlier. Many of you have somehow struggling. So I hope it is helpful for you to understand it. Any confusion, any queries there, definitely you are welcome with that. It's all right? Understood? Clarity came? So I think time up now. So see you in the next class. Thank you very much for your patience-full listening. Sir, can you please go for quick recap, sir? I think another teacher is waiting. So I just told you some moral of the story. It's my pet word, moral of the story. Moral of the story is a DNA first wrapped with histone protein octamer. After wrapping, the structure which form is called chromatin. When this chromatin further condense, it form a structure called chromatid or chromosome. So in one chromatid, always one DNA is there. In a chromosome, what happened? After DNA replication, one more sister chromatid form. See now, here it is a chromatid and here it is another chromatid. These are sister chromatid joined with the common centromere. So we are telling these are forming in S phase. Actually, you think like this is a structure present in G1 phase. And after DNA replication in S phase, this is a structure present in S phase. So in S phase, one more chromatid is forming, and that is called sister chromatid. But because both chromatid is joined with the same centromere, we cannot say it is what? Two chromosome. Why? How can you say two chromosome? Until unless they will not separate. Though I join na, how many pen is there? I am just telling you, how many pen is there? Tell me, count the number of pen. One. I am breaking it. And the pen is joined at one point only. It's not totally separated. At that time, I ask a question, how many pen is there? You will say one or two. You will say one or two. One only. Because it joined, still joined at one point. When it totally separated, it will tell how many. It is totally separated. You will say how many pen are there. It is going to break into two pieces, two pen. Same happen na. This is and this is joined here with the centromere. Until unless it is not separated, as long as it is joined, you cannot say it is 1 or 2. You don't have right to say 2 chromosomes. Are you getting the idea, understanding the idea or not? Even 99% separated, 1% joined, na? So when it becomes separated, like this, you will say, sir, it is 2 now. Picture came in the mind? Understanding came in the mind? Okay, ji. Thank you very much for your patience for listening. Bye-bye. See you in the next class. Thank you. Thank you very much.